Good afternoon and welcome. And I want to thank you very much for actually joining us to this ceremony of the Reverend Joseph Carrier Science Medal. That is the highest honor that the College of Science grants. I'm Santiago Schnell, the William K. Warren Dean of the College of Science, and we're delighted to host this prestigious event now in its third year to recognize an internationally renowned scientist. Before introducing our distinguished recipient, I would like to share a few words about the legacy of Reverend Joseph Carrier, after whom we named this award. Father Joseph Celestine Basile Carrier, CSC, is the Notre Dame first through advocate for science education. He was a brilliant scholar who excelled in science and mathematics. He was born into a prominent French family, educated in the College of Hellel in Paris, and taught natural sciences there. After joining the Congregation of the Holy Cross, he served as an army chaplain during the Civil War before appointed by our university founder, Father Sorin, as the head of the Notre Dame Science Program. Father Carrier's contributions laid the foundation to what has become one of the country's leading programs in science. Of the Nobel Prize Nobel laureates, among its alumni and the former faculty, 2 were affiliated with science. Of the former, current, and recent emeriti faculty and alumni, 13 are members of the National Academies of Science, Engineering, and Medicine in the college. Father Carrier organized courses in botany, established botanical gardens, and founded what is now known as the Notre Dame Museum of Biodiversity. His vision for science education and research continues to inspire the work that we do here today. The College of Science awards the Carrier Science Medal to scientists whose background-breaking research has fundamentally advanced our understanding of the world. Their discoveries are not only transformative in their fields, but also reshape how science is taught. As well, their work becomes incorporated into textbooks and curricula, enriching the knowledge that's passed to future generations of young scientists. Past recipients include Nobel laureates like Thomas Soloff, who won the Nobel Prize in Physiology and Medicine in 2013, and Donna Strickland, who won the Nobel Prize in Physics in 2018. Both who have profoundly influenced how we understand and teach science today. It is now my pleasure to invite Dr. Steve Corcelli, the chair of the Department of Chemistry and Biochemistry, and Father Robert Dow, president of the University of Notre Dame, to the stage to introduce our recipient and present him with the Reverend Joseph Carrier Science Medal.
Thank you, Dean Schnell. As chair of the Department of Chemistry and Biochemistry in the College of Science at the University of Notre Dame, I'm honored to introduce our recipient, Dr. Michael Levitt. As chair, the Reverend Carrier Science Medal is the most prestigious award given by the College of Science. It represents a pinnacle of scientific excellence achieved during years of a career dedicated to discerning the answers the universe has to offer. As we know, science is not static. Ideas are refined and adjusted many times over. Our scientists at the University of Notre Dame embark upon these challenges every single day. Today, our winner has proven how computers, chemistry, physics, and biology can combine to yield world-changing discoveries. The 2024 Reverend Joseph Carrier CSC Science Medal is awarded to Michael Levitt, a scientist whose research led him to be awarded the 2013 Nobel Prize in Chemistry. In the 1970s, Dr. Levitt, with his collaborators Dr. Martin Karbliss and Dr. Arya Warshel, successfully developed methods that combined quantum and classical mechanics to simulate chemical and biochemical phenomena using computers. These computers used punch cards and were the size of entire rooms, yet were substantially less powerful than the phones we have in our pockets. By visualizing the structure and dynamics of molecules, the trio that included Dr. Levitt and others revolutionized the fields of chemistry, biochemistry, materials science, and biology, leading to new insights for the discovery of novel functional materials, the fundamental function of biomolecules, and ultimately, the molecular basis of diseases. Dr. Levitt, who is the Robert W. and Vivian K. Cahill Professor of Cancer Research at Stanford, was born in Pretoria, South Africa. Before his doctoral advisor, the Nobel laureate John Kendrew, would allow him into the lab for doctoral studies at Cambridge, he required Dr. Levitt to spend a year at the Weizmann Institute of Science in Israel. It was something Dr. Levitt was initially confused about, but later on realized that he needed those connections to be successful, and at the time, he needed Israel's superior computing capabilities to ultimately learn to simulate the behavior of complex molecular systems. After Dr. Levitt completed his postgraduate studies, one with Shinora Lifston at the Weizmann and another with Francis Crick at the Salk Institute, he returned to the Weizmann Institute in 1979 before ultimately beginning work at Stanford in 1987. In addition to winning the Nobel Prize, Dr. Levitt is also a member of the American Academy of Arts and Sciences, a member of the U.S. National Academy of Sciences, and a fellow of the Royal Society. On behalf of the University of Notre Dame and the College of Science, it is our great pleasure to award the 2024 Reverend Carrier Medal to Dr. Michael Levitt. Thank you. Now, we'll ask Dr. Levitt to present his talk on multidisciplinary revolution in biology and artificial intelligence. After about 35 minutes, he'll be joined by Kate Bieberdorf, Notre Dame's professor for the public understanding of science, for a fireside chat. Although I don't see a fire, but it will be in that format. And after that, he's going to answer some of your questions. So we really appreciate it. And without any further ado, thank you.
Thank you so much, Steve. I guess the microphone works? Yeah. So it's really an incredible pleasure to be here. I have never been to Notre Dame University before, and the last 24 hours have been really enlightening. I actually feel that I've discovered a new place on earth, new culture, amazing architecture, people, museum, and just completely different. So I'm really excited that this happened, and really, really happy to be here. So my talk has a sort of a mouthful of a title. It's sort of basically everything combined together and sort of my view on how things have been. That isn't very informative, and it'll get better as we move on. So essentially, it's going to be about learning from three things: biological intelligence, BI, human intelligence, and I'm going to call that sometimes HI and sometimes CI, because it's not really human intelligence, it's cultural or collective intelligence. We're much smarter than we used to be, not because we've individually changed, because of what culture has done for us. And then, of course, AI. So let me just start with BI, because in some ways, biology is the smartest thing we know. Everything you can see that is living was created by biology. And again, I'm not going to be theological here. Biology might have had some help. The important thing is that what created biology was not us. We didn't design the flowers. We didn't design birds. We didn't design human brains. As such, it's almost like something constructed by an alien intelligence. And therefore, what really, really matters is learning from it. So biological intelligence is an incredible force for learning. I'm just going to try to say, and again, just showing you that biology can make really cool materials, information structures like DNA, proteins, ribosomes, cells, neurons, hands, eyes, brains, cities. It's all basically through biology. And biology has done this by evolving from very simple organisms way back, 3 billion years ago, for the very simplest organisms. And as things have gone along, we've become more and more complicated. So firstly, it's an interesting question of why did we become more complicated? I mean, what is good about complexity? Why do we need intelligence? Why weren't bacteria intelligent and human beings, very good at sitting in gutters? So the direction of evolution is interesting. And the other thing is that there was a marked change in how evolution works. There really are, in that complicated slide of life, 3 different kingdoms of life. The big one is the kingdom of eukaryotes, and these are organisms like ourselves where every new cell, new species or new baby has 2 parents. Then we have archaea. I'm not going to say too much about them. And then we have bacteria. And bacteria came first, and bacteria basically practiced survival of the fittest in the sense that a father bacteria would make offspring that were clonal copies of it, replications of it. So basically, if you were a very successful daddy bacteria, you make kids who are all like you. And that seems like a really good idea, but it presupposes that the future will be like the present. If the future is like the present, that is a really good strategy. Unfortunately, nature discovered that the future is full of surprises, and therefore it had to make a new step. And this new step was the invention of 2 parents. Maybe, if you like, the invention of sex, where you had a male cell and a female cell. And the key step is that their offspring were a random mixture of the mother's genes and the father's genes. It wasn't like the best from the mother and the best from the father. It was to try to keep things as diverse as possible. Nature made that big step forward about 1 billion years ago. And everything you can see around us, grass, trees, birds, everything, flowers, people, all came from organisms that have 2 parents. So it was basically the creation of offspring who are not your clones, but only 50% identical to the father or the mother that led to this massive burst of life on Earth. So essentially, if you think about this, it's not evolution or survival of the fittest. It's survival of the most diverse. And this sort of makes sense. A good banker, I'm sure Warren Buffett would agree with this. You basically don't buy one stock. You buy a diverse portfolio of stock. And the chances are some will succeed and some will not succeed. But on average, you'll do better. In exactly the same way, by having a mixture of what went before, it was better for survival. It also explains why things got more complicated. Because a complex organism has more different kinds of behavior than a simple organism. So a flower is complicated, maybe even has bigger DNA than I have, but basically it doesn't have a complexity of behavior. It's stuck in a pot or stuck in the ground, it can't move around. If the weather's bad, it can't go inside, it can't turn the air conditioner on. If you think just how complicated our behavior is, it is amazingly complicated, and it's this complication which we call intelligence, but again, higher animals are also very complicated, that enables us to have much greater potential for survival in the future, unless we destroy ourselves, but let's hope we don't get there. Okay, so let me just say now something about HI or CI, human intelligence, and talk initially about 2 things, great scientists and great institutions. So great scientists serve as role models. Role models are really, really important in science. You want, in some ways, and it's not easy, to have people who have been recognized for whatever reason to act like rock stars, to kind of drive around in fancy cars and maybe do their hair in a certain way so you'll recognize them. And because they're meant to be a symbol of science. And for young people, you want to attract them into science, and therefore acting in a preposterous way is probably a really good idea. I'm not very good at that, although my wife might say I'm actually very good at that. But outreach is very important. Now, I realized this basically a few hours after hearing the news about the Nobel. And I said, well, I'm exactly the same as I was 3 hours ago, but why are all the newspaper people here, and why is all this fuss? And then I suddenly remembered the high regard I had for other prize winners when I was 20. When I was 20, the way Francis Crick behaved was incredibly important. Even the way he misbehaved was really important. And I'm not going to go into that right now. But there was all this thing, but they were human beings. To realize that these heroes of science are actually just normal, fallible human beings. who have dislikes and likes is a very important lesson. It suddenly makes it all seem much more approachable. So basically, my heroes of science helped reveal the basic secrets of life through the understanding of the intimate details of how biochemistry works. And this group was a small group of people based in a small laboratory in Cambridge called LMB, or Laboratory of Molecular Biology. And I actually appreciated them in 2012, which was before I was in any way recognized. I had to write a piece about them. And I basically called the article My Towering Heroes of Science, as an appreciation for people like Max Perutz and John Kendrew and Francis Crick, who have all been mentioned before. So it really was a very important step for me. Let me just go through these people. So first is Max Perutz. He was the person who founded the lab. He was a Jewish refugee who had come from Austria to Britain. Actually was interned by the British, but was always very creative and founded the lab. And besides inventing protein crystallography, which enabled us to see all the details of these amazing molecules, he had the best empathy and the greatest emotional intelligence of everyone I'd met. And this will come out a little bit during the talk. The next person was John Kendrew. He actually got me into the field because he had a television program on. I came to England from South Africa, where I was born at the age of 16, became immediately addicted to television, could not tear my eyes away, and fortunately, he had a program, an introduction to molecular biology, on the single channel of the black and yellow BBC television that I saw. In those days, Scientific American was really important because the best papers went to Scientific American first. It was the only place that published in color. And if you had solved a structure as complicated as that, you really wanted it to be in color. And what was great was that 15-year-old boys were reading Scientific American. It was a journal that was open to people. So it was, again, something that was very accessible. Third is the scientist we probably know best, Francis Crick, the co-discoverer of the DNA structure with Jim Watson. Crick is famous for being so smart, but I want to tell you a different thing about Crick that maybe people don't realize. When he was working with Watson on the structure of DNA, he was actually Max Perutz's Ph.D. student. Furthermore, that fuzzy picture you see in the top right corner of the map is a diffraction picture that Perutz had obtained from Rosalind Franklin that allowed Crick and Watson to solve the structure. So Perutz was the supervisor, played an important part of the discovery, but he is not mentioned in the paper at all. He is not thanked. He is not a co-author. Now, can you imagine if you had a student today who has just done the most important thing you could imagine, and it was very clear that it was very important, deciding, no, he should publish by himself. I'll help him publish because I'm sure that Perutz got it into Nature. But he basically believed in one key principle, and that is you need to make young scientists independent and responsible. And therefore, if the model was wrong, it's not Perutz's doing, it's Crick and Watson, and that's the right approach to take. Anyway, these heroes of mine basically solved basis of biology, and you can really summarize all of biology on one slide. You have DNA that tells the cell how to make proteins. The proteins fold up into certain very precise three-dimensional shapes, a bit like Chinese knots, but not really knotted. These shapes then have a function. In this case, they recognize a food molecule. They metabolize the food molecule. That's good for the cell, so you make more of the DNA. It's a very simple idea, but it's a way of learning. I mean, nature has slowly learned, and in some ways, we are our history. We contain in us all the solutions that nature has found. Another reason why things get more and more complicated, because we keep on taking genes from different places and adding them together. It's not a competition. It's A and B, not A or B. Just a picture of DNA. DNA is incredibly important. It's so tiny and compact. We talk about our best printed circuits as having 5 nanometer scale. That piece of DNA is 2 nanometers across and already contains all the information. Just the wire in the best printed circuits is 5 nanometers across with no information capacity at all. So this is really high information, self-correcting, and a code. Perhaps more important than the DNA are proteins. There are also necklaces of 20 different groups. I should just go back and say that in DNA, the different colors you see there on the right are indistinguishable. They differ by 1 or 2 atoms. They're very, very similar, the base pairs. But that's enough to distinguish the code. Proteins actually have groups that have much more shape to them, different amino acids. Some are big, some are small, some are positively charged, and so on. Now, this chain of amino acids does something really amazing. It self-assembles. It folds up into a compact shape that actually has very precise three-dimensional structure. This is an enzyme, this is an inhibitor that stops the main enzyme in our stomach from digesting ourselves. We're made of proteins, so you have to control. This is an inhibitor, a very small protein. But in our bodies, we all have many of those, and they all look just like that. So the shape is precise. So now we have microengineering at a scale that's unprecedented anywhere in the real world. We still have to get there. Okay, so one other thing about great institutions is if they're run properly, they can win many prizes. And Perutz really had a genius for making this happen. So he had a genius for what he did with Crick. He also was, John Kendrew was Max Perutz's assistant. But when John Kendrew published his papers, they were in competition with Perutz, not under Perutz. So again, the idea was you want to have as many bright fires burning independently to really create as much innovative science as you could. I got to that lab in 1968. It was fairly early, but still, and I was very happy to get there by going via Israel. That was important. So what is the secret of this institution? And, you know, different places are very different. I just know this place. It's a long list. They've won 28 Nobel Prizes in 60 years. That's more than Japan. And the actual population of the people who were there as students, postdocs, or whatever, is probably 3,000. So it's like 1 in 100, whereas Japan is 100 million. It has the same number of prizes. And some of these prizes are DNA sequencing, protein sequencing, monoclonal antibodies. And you know, you can just go along the list, cryo-EM, crystallography, DNA structure. It just goes on and on and on. And they've done this over the years steadily. So it's a kind of an amazing contribution. So you could ask very simply, what is their secret? And I try to say a recipe for Nobel Prizes. It's making a bit of a joke. So I think first and foremost, make young scientists independent and responsible. As a beginning PhD, and I studied physics, I didn't know any biology and any chemistry, but if somebody like Max Perutz didn't feel like going to a conference, he would send one of the PhD students. Now, I think it's maybe not fair for the conference organizers, but I guess he was big enough to do this happen. And if you're an undergraduate or a beginning graduate student, you get sent to a conference where everyone else is really distinguished, you really work hard, and you really feel committed. It had small groups. Small groups make great sense because no one really knows what's important in fundamental basic science. One thing you can be sure of, if somebody says, I know what it is, it's almost certainly wrong. And you don't know, just the odds are. So they would have small groups. When I was there was no such thing as a grant. A group got funding. You also had unlimited access to chemicals, to computers, to computer time, to the machine shop, and it didn't really matter. You didn't need a requisition. Perutz used all his smarts to eliminate bureaucracy. He felt it was really important that young people shouldn't have to waste their time teaching, forgive me, filling in forms, teaching, having a group. They were meant to be working on their own thing, and it was very common to have people who were working at their own bench for 30 or 40 years just because they wanted to do the science. So I think there's a recipe there. You know, early independence and responsibility is the secret of Nobel Prizes. Now, everyone thinks that people like me, you know, you get recognized when you're really, really past it all. But you do the work. In my case, the work was all done for me by the time I was 25. Now, I didn't know that, and I don't think I would have known and said, okay, I'm going to retire now and just wait 40 years and so on. But it shows you that the real emphasis is on young people. And I worry these days that as people are living longer, as people aren't retiring, as the baby boom is moving through society, we're having too much emphasis on old people and not enough emphasis on young people. And I feel this very, very strongly, and I actually tell people, you know, look around at what's really changed the world in the last 30 or 40 years. Steve Jobs, Bill Gates, Sergey Brin, Larry Page, Mark Zuckerberg, and others. In other words, OpenAI, Sam Altman, otherwise people who could not wait to finish their education had to drop out of a Ph.D. or a B.S. program so they could change the world. And I think there's a really important message there. And these scientists used to be in that role, and it's not happening so much anymore, and we have to ask why. And I think it's basically because people are getting older. Now, there's nothing wrong with getting older, but your role when you get older is to really help young people, and we need to realize that. Okay. So let me just now say something about my own work. I've called it AI, but I could have called it HI, because you could actually argue that anything you do with computers involves AI in some way. AI as a field has been around for a very long time. Before my PhD, there were groups working in AI in the 60s. They just didn't have the computer power. So essentially, my own work has almost always been basic science in computational biology. I started out with a detailed model of tRNA. It wasn't really important, except that Crick got it into Nature with me as sole author in 18 days. And I only realize now in retrospect he was probably paying forward what Perutz had done for him for the DNA paper for 16 years before. I never noticed that, but I did thank him. My own work in Israel, I actually met Lifson, as Steve has said, and worked with him on how you could put a protein into a computer. I started out working on simple small molecules with Varshil, who was working on small molecules, and very quickly we graduated to protein molecules, and we found ways in which really weak, tiny computers with a memory of maybe 200K could actually do something useful, but it was possible. Then I went back to England, did my PhD there, came back to Israel, and with Varshil did a simplification of proteins where we said the important thing is that they're chains of beads. The atoms don't really matter so much. Therefore, if we simplify them, we have enough computer time to simulate their folding. This is way back. Then Ari and I also did some work on enzyme action, again saying we need electrons, so let's make things more complicated. It's not, but these are all very early papers. And then something I did in 1979 was to actually make the first movie showing how proteins move. Movies back in those days were really, really hard. This movie was made at NIH on a special purpose computer. Each frame of the movie was like $1,000 of compute time. And you have, 25 frames a second or something like that. So they made this for me, and it's the first movie showing how atoms move in a protein. And there it is. And I've seen this now like a million times, and it doesn't, but I remember at the beginning being completely flabbergasted because it looks like a bag of worms. I mean, they're all moving. They're not really going anywhere. They're not moving, and this is probably how proteins really look, and I think now dynamics has become very, very common. Of course, besides just moving, that's a water molecule coming out, and back at that time, we were very concerned about the space program and things like that. Enough. Okay. More recently, I've been looking at functional motion in my own work, and this is just a movie of showing how the ribosome, which manufactures proteins from DNA through RNA works. It's not really important, but the idea is just to take these very large structures and essentially make movies. It's a bit like taking still pictures of people running in the street and then trying to work how you run, putting them in the right order till you see the motion. Okay. So I also did applied work. I was actually very interested, almost accidentally, in doing applied work. Shortly after getting to Stanford, I had been doing modeling of antibodies, and this led to applied work involving antibodies. This is in 1987. It's important to realize that we couldn't even think about cloning proteins until 1986 or 87. So it was a very early time. We could actually make proteins. And the idea of antibodies, and we all know about antibodies thanks to COVID, these are molecules, they're just proteins. Each protein is a chain of 120, and it makes something looking like a hand with loops, and these come together, and the loops have different shapes, and it's the loop shape that recognizes what's going on. Could be hands like this, but they actually bent back. Okay. So basically, in 1989, with a company called Protein Design Lab, we wrote a key paper called a humanized antibody that binds interleukin-2 receptor. So interleukin-2 receptor is a protein expressed a lot on cancer cells. So if you have a cancer cell, that is, you can recognize it by looking for interleukin-2. Antibodies do recognition. That kind of makes sense. So if you had an antibody recognizing interleukin, you could perhaps inject it, and it would be good therapy because it would get the immune system to kill that cell. Problem is humanized. Difficulty is the way you make antibodies, to take the interleukin-2 cells, inject them into a mouse or a rabbit. That raises a response. You extract serum, purify it, and see what the protein sequence is. And now you can make the sequence. Trouble is, you can make the protein, but if you inject a mouse antibody into a person, the person says, no, that's foreign antibody. Maybe it's making me better, but it's also making me really sick. So humanization is like a makeover of the antibody. You want the antibody to sort of act the same, but not look foreign to a human cell. And there's a trick for doing this. And you might argue, well, just take the fingers and put it onto a human hand. It'd be the easiest thing to do. And that was actually proposed early on by Greg Winter, who also actually won a Nobel Prize from Cambridge 3 or 4 years ago, where you would take the gray shape that looks like a Pac-Man and replace its red lips by what comes from the mouse. And then it should be a chimera of mouse variable loops and human framework. So most is human, but the bits that do the action are not. Problem with that is that these pieces have to have the right shape to recognize the interleukin receptor. And if you mess around how they touch the gray part, things go wrong. So the real clue is shown on the right, and this was what was sort of in that paper at Protein Design Lab. You have to also change a few amino acids in the blue region. A tiny change wouldn't probably even merit a paper, but it got a patent. And the patent said, if you want to make antibodies humanized, you've got to make some changes to the gray region. This ended up meaning that what if you did what Winter said, it wouldn't work. And it's kind of obvious, if you make more changes, obviously you're going to be better. But that doesn't matter for the patent office. This patenting was actually very important because developing antibody drugs is a very expensive process. The company Genentech in South San Francisco spent literally hundreds of billions of dollars developing methods to humanize, to produce the antibodies. These are not pharmaceuticals, they're proteins, they have to be shipped in the right way, they have to be injected and so on. And they wouldn't have done all this if once they developed the whole method, anyone else could just do it. So they were very happy about the patent. And this led to a number of therapeutics coming from that patent, drugs like Herceptin, Avastin, Lucendia, and so on. And these drugs actually still used today. They're all off patent, which is probably a good thing because the patent expired in 2014. But also the company earned more money from the patents than from anything else it actually did. So patenting is important because it gave the chance to develop. Let me talk about something else. And this is my more recent work in computational biology, and it involves COVID. And basically, for pure chance, and chance is a very important driver in one's life. My wife sitting here is a curator of contemporary art in China. We'd been visiting China in the fall of 2019. We had friends there. They were really upset when the pandemic started to spread in late January 2020. And my wife wrote them a very nice message. And they loved it. The response was like, wow. So when we got it, and it made me realize, gee, they're really upset. They're scared. So maybe I should look at the numbers. And you know, my real love are computers and numbers, and it doesn't really matter what the numbers are. I'm happy to work on any numbers, but I just like data analysis. So basically, this led for me to write a couple of reports. Well, the first report came out on 2nd of February 2020. Back then, we knew how many cases and deaths there were for, I think, 10 days, very early on. But when you actually, and this is the second page, it's not important, but basically, what you actually see, if you want to know if something's growing exponentially, say your bank account, they tell you they're paying you 2% a day, very generous bank. What you would do is you would take the amount you had today, divide it by the amount you had yesterday, and say, did it increase by 2%? And then maybe not believing the bank, you would try it the day after. So simply, the ratio of the total number of deaths or counts or cases today divided by yesterday should be a fixed number. If things are increasing at constant exponential rate, it should be a fixed number. But it turned out, looking at the data from China back then, that the number was actually decreasing. It was really rapid at the beginning, but dropping very quickly. So that looked hopeful to me. I didn't know any epidemiology. I didn't know anything about this. But as looking at a curve, you see a curve moving down. Maybe it's good. But I was actually hooked onto this because my first report was leaked by somebody in China. It was translated into Chinese and viewed by 5 million people within a couple of hours. This all happened when I was on a plane on the way from Israel to New York, and I landed, and there were all these emails saying, "Are you the Michael Levitt from Stanford who wrote about COVID?" And I said, yeah, I didn't publish anything. I sent it to friends and whatever. But it basically meant that I had to continue, and continue I did. And the important thing is the very top curve on the right is showing it going down on the 2nd of February. If you extended it, it just keeps on going down. It doesn't actually go down as a straight line. It drops exponentially, as we now understand. But basically, for some reason, in that very first pandemic, it seemed to have built into it a slowing down. And we didn't understand this, although slowly, in collaboration, the understanding came. So essentially, there's two kinds of growth. One is growth where basically the number of cases per day goes up and comes down in a symmetric way. That's like a Gaussian function or a logistics function. And in that case, if you took tomorrow divided by today, it would always be the same number until you got to the very end. So things are growing exponentially until you exhaust the pool of people. And then, of course, it has to stop, because you can never grow exponentially forever, because at some point you stop. But the important thing is that today to tomorrow is always the same, whereas the other curve grows very, very quickly. The curve at the bottom ends the same way, but it starts off growing much, much more quickly than exponential, but it's slowing down all the time. And for that curve, you can actually tell when it's going to stop, and that's important. Okay. This just shows that a lot of the actual curves that occurred during the pandemic look like that. The very earliest ones, in that case, in New York City for cases and deaths, something towards the end of the first year. I think it says Ireland. Then China got infected much, much later, Hong Kong and then Shanghai. And again, when they got infected by the same mechanism. We say this because you don't know whether things rise very slowly at the beginning, because you're not measuring cases. But there's no doubt that places like Shanghai and Hong Kong, people were monitored. They had to do a COVID test every single day, whether they traveled or not. So they really knew what was going on. So then, together with a colleague, actually at a university in Shanghai, but an Italian from Padua, we had an idea that what would cause the spreading to be very fast at first and very slow afterwards is the nature of social networks. So epidemiologists assume that the network is like the chairs in this room. Every chair is surrounded by 4 chairs or 9 chairs or whatever you like, but every chair is the same. But if you take social networks like Facebook or Twitter and say, how many followers do different people have? Some people have lots and lots of followers, but most people have very few followers. And the same thing seems to be true of a human network, where some people, maybe because of their job, maybe because of where they are, have lots of contacts, and other people have much fewer contacts. And the important thing is that you have what is called a scale-free network, where the number of contacts is distributed in such a way that there's quite a good chance to have very many contacts. So basically, it's a power law rather than a Gaussian. And this, if you have a law like that, and you do a simulation of infection, you would get exactly the Gompertz function. Basically, at the beginning, you infect people who have many contacts. They're easy to infect because they have many contacts. Once they're infected, they then infect many other contacts. But after a while, you use them up because they're no longer infected, they're cured or dead. And basically, when you do this, you get a picture like this. You can actually simulate what this looks like. It's kind of hard to see, but you can see red areas appearing. Maybe those of you who are close to the screen, there's like a red patch, and it's growing by this way. And essentially, what you're seeing in the growing curve is a line dropping. That is the today to tomorrow. And today to tomorrow should be a straight flat line, but it's a decreasing line just because you're running out of the super spreaders. Okay. So basically, that was interesting. This just says the same thing. Let me just talk a little bit now about the other side of this. One side of this is the dynamics, and I was naturally attracted to the dynamics because of dynamics of proteins and things like that. But there's another very important side about a pandemic, is just how many people are being affected. And that turns out to be a very, very difficult number to ascertain. It should be really simple. Take some typical years, compare the amount of death during the pandemic years, and see is it up or down. And unfortunately, it isn't as simple as that. I think that it could be as simple as that. We decided to take as typical years, 17, 18, 19, just the 3 preceding years. It turns out that in some countries they were very good years, and later on we tried methods that would take all the different years together. When you do this actually led to a paper and another paper. Basically what you find is that, and again it's really, really hard to see, this just shows how much extra death there was as a percentage of normal death. Here shown just for the 2 years of 2021, and basically, I can see it over here, the very top line is Poland with 15% extra death. The next line is the United States with also 15%. But if you go down, say towards the green part, the green part are people who have 8% extra death a year. 8% death is an extra month of death. And one way to think about death is that the angel of death has decided to come to you a certain amount of time sooner. And that's particularly true when the age profile of those dying is the same as the natural death profile. If it was happening to 5-year-olds, if you suddenly had an extra 15% of 5-year-olds dying, you'd be really, really scared. But of course, very few 5-year-olds die. And if the number of extra deaths is proportional to like the normal deaths, it means you're just bringing death forward. And it also means that if you die from COVID, you won't die from the other death you were going to die from 3 months later. So over the time it ends up, and you even find at the bottom of that table, the very lowest country is New Zealand and then South Korea. These are countries where they actually had less death during the pandemic than in the 3 years before. Why? Why would they have less death? It turns out, for reasons that are still not understood, flu went away completely during the pandemic. And if you were a country that had high flu deaths and not very high COVID deaths, it went away. So again, kind of a sensible story. Just finally, I think this is my almost final slide. If you compare United States and Europe, United States at this period have about the same amount of excess death, 700,000 versus 600,000. The big difference was that in the United States, there were many more deaths of people under 65. For reasons that we now understand, but we didn't then. Just my last slide in this section, just showing that it doesn't matter what reference years you use. The United States had a lot of excess death compared to previous years. Sweden had less, and so on. There's a whole spectrum of what's going on. One slide that I forgot to include here was if you plot the amount of excess death against socioeconomic indices, the one that really stands out is inequality and poverty. And for the United States, it's really striking. The amount of excess death in different states during the whole pandemic has a 0.87 correlation coefficient to percent poverty. So basically, places that had poverty had very high excess death. And I think the ratio, the worst poverty place in America is 3 times higher poverty than the best. But COVID deaths did exactly the same scale. So let me change subject a little bit to something perhaps more encouraging, but scientists don't really care about that. That's AI now, and that is basically generative artificial intelligence, which we're now experiencing. And basically, everyone knows about it now, but for me, these are very important dates. The beginning of December 2022, GPT came out, and essentially I have been talking to it continuously ever since. It records all the questions, so I think I have something like 10,000 conversations with GPT, and thousands of pages of paper. And essentially, it is amazing. It completely blew me away from the very first moment. In 20 months of using AI, I can use it to write computer languages I don't know. I can use it to write articles in languages that I don't know. I can use it to translate people speaking in front of me in any language they like. It's great for old codes. I have programs that I've completely forgotten why they were working. Can you please explain it to me? It's good for that. Medical advice, legal advice, psychological advice. It's really, really good for these. Essentially, I haven't asked it a question where I haven't been impressed by the answer. And the answer wasn't always right. The answer was sometimes wrong. But actually, that didn't bother me at all, because even my smartest friends say things that are wrong. It's perfectly okay. You know, I think somebody who never made a mistake would be doing something really easy and boring. And I think this is a great thing to worry about. And I think it's true of all walks of life. I think people who are working in an office, they need to try innovative things so they make mistakes and not fear for their jobs. Unfortunately, that culture has also changed a lot. Okay. Two areas of GPT really impressed me: emotional intelligence. So this was a preprint of a paper done, I think, at Tsinghua University, where they actually tested the emotional intelligence of 500 adults. I argue they were probably all engineers at Tsinghua University and therefore have low emotional intelligence, but GPT was in the 90th percentile of that group. So that's actually, and again, if any of you don't believe me, just go and ask it an emotional intelligence question. Anything. I mean, it's all private, and you'll be amazed at how smart it is, because it's giving you balance. Maybe you could find the advice, but when you want advice, you're not going to spend an hour searching for it. You want to tell me what to do. And another thing that was a big surprise is that there's a board game called Diplomacy, which hardly anyone knows about, but some do. It's a map of Europe in 1901, and you have to make alliances to prevent World War I. And basically, the game is to simply say, people get assigned by dice random places in the screen, and now you have a quarter of England, and you have half of Germany. So now you'll maybe say to somebody who owns England, I'll give you a quarter of England if you give me the northern Germany, and so forth, try to build complete countries. Turns out that AI, which actually was published before GPT, from Facebook, from Meta, their group, published AI called Cicero that is basically the world champion at diplomacy. But more interestingly, people who lose don't actually enjoy losing, because he doesn't cheat, he doesn't stab anyone in the back, he just understands the strategic significance of different places in Europe better than any human being. So that, I think, is also very exciting. So, for me, just quickly to end, I have a vision for education that we all need to embrace AI as a smart assistant, whether it's for cooking, or what clothes to wear, or maybe a new tie, or anything you want. You have a day in Rome, what should I do in a day in Rome? You love eating a certain food. It is really, really helpful for these things. Again, the information might all be there, but it puts it together in a very, very good way. Essentially, AI is greatly increasing cultural intelligence. It isn't making my IQ necessarily any higher, but if I've got my smartphone, and my smartphone is connected to smart AI, I am much, much smarter. And one thing that I think we need to also prepare for lifelong learning. Essentially, we have to keep on learning, and this thing makes it really easy to learn. So for example, let's just imagine I decided to retire, and as a hobby, I'd like to buy a yacht. My wife would not like me to do that, but I could probably learn all about yacht ownership in half an hour using ChatGPT. And it would tell me where I should go and what I need to do, etc. And I've used it for countless problems like this and being very impressed. What I just wanted to say in ending is that an amazing thing is that 80% of the world touches a smartphone. Maybe not an iPhone, maybe one made in Korea or China or hand-me-down. This is an amazingly big number. 80% of the world probably don't eat breakfast every day and probably don't sleep in a bed and almost certainly don't wash every day. So 80%, the iPhone has become the most democratic, or the smartphone, the most democratic thing in the whole world. And this is going to get smarter. It's connected to the internet. It's going to get smarter and smarter, and people will get smarter with it. So, closing words. So I sort of see that we can summarize what evolution has taught us in one slide. 3 billion years ago, we had a bacteria. I'm calling it a man bacteria, although my wife says I should call it an it bacteria, but it's kind of stupid. Simple, fittest surviving, that's sort of chest banging. 1 billion years ago, we had male and female. Suddenly, we had complex, diversity, and thriving. Everything we see around us is a result of that invention. And today, I see we are actually adding an A to our BC, and basically adding AI to the triad, and having humans, biology, man and woman. And this will lead, I think, has a very good chance of leading to a really hyper-complex paradise. Finally, I just wanted to acknowledge Max Perutz. He said, in science, truth always wins. And I really believe that, and it's something that's been with me for a long time. As a joke, and this somewhat relates to COVID, I'm still writing papers on COVID. And I told my wife the other day, I actually don't really care whether people read the papers. They'll be in the archives. GPT will know what to do in 10 years' time. Thank you all for your attention.
was absolutely amazing. Can we give a big round of applause for Professor Levitt here? So we have time for a few questions. Do you want to come over here and join me?
Sure.
Perfect.
Wait.
So
I
I
don't
want
go anywhere without that.
Oh, yeah, you can't do that. Okay, so I want to kickstart our conversation by asking you basically a very simple question. What intrigues you most about computational biology?
I think when I heard from the lecture that Kendra gave on television in 1964, that basically life was little threads, tiny microscopic threads, that folded up into specific shapes that were the basis of all shape in life. I just couldn't believe that. I mean, you know, and if that had been a science fiction story, you wouldn't even bother to read it. It was so nonsensical. But this is what nature decided. Self-assembly is very important because everything that we make has a factory to make them. But life comes from inside. It starts from nothing and makes itself. And to see how this is implemented, but it's still using physics and chemistry. So how this thing we call life, which is quite amazing, used physics and chemistry to create things. And you know, the level of order in a hand, it makes our microcircuits seem really crude. We're getting there, but there's this incredible level of order of things working together. I mean, in some ways, inside us, we're much more like clockwork than chemical mixtures. And everything working precisely, timed, still with lots of randomness, very robust. I mean, a body goes on working for 70, 80, 90 years, and you know, there are occasional problems, but it's basically functioning, and it's extremely complicated.
So now I want to know more about your process. So let's say you want to learn a new skill or you have a challenge. How do you work through a problem?
I think, firstly, I'm interested in almost everything. So that makes it very easy because you keep on seeing things that interest you. And it could be a floor tiling, it could be a wall. And nowadays it's wonderful because you can, you know, a while ago I would go on the internet and find out about it. But often I would ask them to recommend a book, so I still read books. But often, you know, recommended by, basically, for me, AI has become a continual companion. I asked it, please summarize the origin of Notre Dame University very quickly, have it on one page. I mean, you know, it is just very, very convenient. It basically, I think, removes the barrier to entry of all new fields. When you come to a new field, you don't know who the best person is, you don't even the journals, you don't even know anything, what meetings. It can give you the introduction, and not necessarily to the field, but to the content of the field. So I think it's really reduced the barrier to learning completely, and it's not just in the area of science, it's in any area. If I wanted to fix motorcycles, I'm sure I could get advice from it. And the advice, maybe, again, it's not in any way knowing everything. It makes mistakes, but it's basically no worse than asking advice from people, which is, everybody. I should also say, maybe, as a scientist, I think we have a belief which we often don't enunciate. We believe that everything is false until proven true. A good scientist really believes that his graduate students, who he loves very much, are planning false data in his papers to destroy his reputation. And he has to check against that. In other words, he loves them, he believes them, but you have to, and a lot of people don't, and they get caught, because it's very tricky. So therefore, and I think it's this feeling which makes me much, much less worried about fake anything, because I don't expect things to be right. On the other hand, there are enduring scientific truths, which are very fundamental.
You mentioned interdisciplinary work, and your colleagues have always praised you for that. So I'm curious, is there a field you have yet to kind of dip your toe in that you really want to try?
It's all accidental. I mean, I got involved in COVID because I was jealous of the likes my wife got from her friends, and I wanted to do something to get some likes as well. Silly reason, but it got me into it. Now, once you're into something, it becomes interesting. And I really think it's very hard to find something that's not interesting. And people often ask me, what's the next thing to do? I have no idea. I actually tell them, you tell me what I shouldn't do, because even that you can't tell people. Stop working on bacteria, they're no longer interested. Oops, sorry about CRISPR. This is happening all the time.
Right.
So I think that life is really amazing. People are incredible. I'm actually very interested right now in problems in the social sciences. So I think I would, and also in, another really strange thing about human avatars, putting all my information into a machine so my great-great-great-grandchildren could access me. And that's also possible. These are just crazy things. You know, I think being crazy is a good idea.
It definitely could lead you to a couple good things, but it could possibly lead you to bad things. So let's talk about potentially academic freedom and how important that is here. So where are you landing on that these days?
So I feel that these are very, very hard. No one knows what exactly to do. Basically, there are lots of unknowns, and I think we should be open to inquiry. I think that somebody saying, you know, it was actually interesting because I know personally the chairman of Alphabet. He was a former professor at Stanford. And there was a time when YouTube was shutting things down because they disagreed with what the World Health Organization was saying. So I said to him, okay, this is fine, but when the World Health Organization changes his name, are you going to boost those articles again? He said, sorry, we shouldn't have banned you. We're bringing you back, and we're going to give you a bonus of, you know, your lost likes or something like this. And it didn't happen. So I think but I really think it came from a great deal of fear. Very, very early on, I was commissioned by, I think, the Wall Street Journal, but it could have been the Washington Post, and I don't want to get them mixed up, Wall Street Journal, to write an article on COVID. This is at the end of March 2020. And I wrote an article which they didn't really like because it wasn't anti-Chinese enough. And, you know, it was basically anti-COVID. It wasn't, but I felt that their numbers were actually kind of reasonable, and in retrospect, I don't have any reason to disbelieve them. But at the very end of that, I said something which I think is coming true faster than I believed. I said that I wasn't really impressed with human response to the pandemic, but next time it comes along, AI will be sufficiently advanced that we can actually ask our smartphones whether we should panic and believe the answer. And I do believe that might seem preposterous, but if you all think how your driving habits have changed since we had, you know, GPS guidance on our phone. It used to be, wife would say, I think you should go this way. The husband would say, I want to go this way. Family fights about it. Now you just simply do what Google or Waze tells you, and there's no argument. So almost everyone trusts the GPS more than their own macho instincts. Now that should happen for everything. And if we get experience, let's imagine you start to ask AI for advice about dieting. And for whatever happens, it's customized for you and it works great. Yeah, you'll start to believe it. Then you'll ask it maybe for political advice, and, you know, good recommendation. So I think we will build up trust, maybe quite quickly. I think the one area where human beings are really, really deficient is in risk assessment. I read an article once, you know, what are the perceived risks of going to the beach versus the real risks? And the perceived risks are drowning, or maybe sharks and drowning. And the real risks are unhealthy food and sunburn, by far.
Yeah.
So, you know, but I think we need to learn how to do this because basically risk assessment is incredibly important. And again, for everybody. So I see these things happening, and, you know, if I can be involved. But again, what I really like doing is just sitting on my computer, running computer programs and playing with numbers. And I don't really care so much where they come from. Yeah. I became interested in epidemiology and demography, and I have some colleagues, and maybe I'll do things in that. But, you know, I don't know. I'll give you another crazy example, which I should have mentioned. So we were in a museum in San Francisco, and there was a painting of Venice. And we'd been in Venice a few months before, and I sort of said, well, I took a picture of it, and I asked ChatGPT, from this picture, can you tell me where it was taken? And it was hopeless. It couldn't do it. But the good thing for me is in thinking about it, I know exactly how to do it. And it basically depends on the length of the painter's arm and how wide the painting is from his arm. And that's 90 degrees. And from that, you get an angle of the picture, and you can get all the angles. And once you have angles, heights become distances. Very simple. Now, I don't know if that's written down, but I'm the first person to think about that. I doubt very, very much. Every photographer should be completely aware of this. But it was an example. So, you know, we were in the gallery. It took a few minutes. AI is also very good at looking at paintings and telling you what it sees.
Well, we actually put out a call to see if we could get some questions for you, and a lot of them were about AI. So
Good.
Can I hit you with a couple of them here? Here's the one from Nate, if you're in the audience. Hello. Okay, well, this is from Nate. So do you expect a future where machine learning systems engage in productive, autonomous scientific research, for example, by autonomously designing and executing experiments in cloud labs?
I don't know. That could happen, but I also believe that such a system, coupled with a human being, would be better than the system without the human being. In exactly the same way that A plus B, even if B is very small, because oftentimes it's the crazy idea, and basically human beings, I think AI will be incredibly have enormous admiration for human beings that manage to self-assemble. AI got assembled by human beings, but we self-assembled, and that's pretty amazing from very, very, very simple beginnings. So I think, for me, I think I really see AI as a smartphone, as a very, very smartphone. And smartphones have enabled us, most of us would, I don't know what we would give up rather than give up our phone for a month, but probably an awful lot of things. And this would be tantamount to a prison sentence of some kind. So I don't know. I actually feel that the real future is AI. It's like saying, well, do you think a cell phone lifts by itself on a bench could do dangerous things? And I guess it probably could call people.
Yeah. Well, that actually brings me to a question from Raymond. Do you believe computers will ever be conscious?
So I wish I knew what consciousness was.
That was the second part. What is consciousness?
So it's a feeling that I have about myself, and I certainly think I'm conscious. But anyone else, I kind of guess they're conscious because they seem to behave more or less like I do, but often very differently from I do. Certainly between men and women, I think most men have a hard time guessing how a woman will behave. You can do it maybe with a lot of practice and a lot of time. So, and I think we do construct models. Part of the way we think about things is we do construct models of other people, because you don't want to stick your foot in your mouth whenever you open it. You need to have some idea of the likely response. But I think that, I think consciousness is a very hard question. Again, I am not sure what consciousness is. And it may be something, I should say the following. I'm actually amazed that writing can be boiled down to such a simple calculation as is done by these large language models. In other words, you can, just by predicting the next word in a short string with a few other catches and a few iterations, you can actually write really, really well. Now, I'm not sure I like the writing style, and it can make poetry and whatever, but it's not maybe the best of human beings, but it's certainly probably it's better than the average human being. That's pretty amazing. It can translate way better than the average human being. And all of this is done by guessing the next, so maybe a lot of thinking just comes from guessing the next word. So I started to think about, is my intelligence just because I've encountered lots of things and I can guess the next word? I don't know. I think we may learn more about consciousness from when we have machines that are completely indistinguishable from us, and then what is consciousness? And I don't know. I think there's been a lot of discussion in general about AIs, whether we have AGI or artificial general intelligence. And certainly for many of the tasks, ChatGPT has for me AGI.
I'm being told we have 1 minute, so this is your 1 minute warning, sir. Last question. This is from Natalie Sargent. She's our Director of Faculty Recognition. Don't know if you're here, give a little wave. The gist of the question is essentially, what advice on persistence would you give to early career faculty and students? And you're now to 45 seconds.
Okay. Love what you do, have passion, don't be deterred by adversity, and be as open as you can to new opportunities and new areas. Basically, act like a 7-year-old child for as long as you can.
I love that. Let's give him a huge round of applause. Thank you so much for your time. Amazing.
Well, thank you, Dr. Levitt. Thank you, Dr. Bibbendorf, for your insightful conversation. I really enjoyed it. Please join me giving one more round of applause to our 2024 Career Science Medals. So now we want to invite you to continue the celebration at our reception located just behind you, where you can enjoy an evening of excellent food and drink. You know that Notre Dame never fails at that. You're also welcome to step on the terrace. And It's a beautiful afternoon, late evening, so early evening. So you can take a view of the campus, enjoy it yourself, take some memorable photographs. And thanks once more for joining us in this ceremony and in this evening. And we hope you enjoy the rest of your night. Thank you.